Testing, 1, 2. Testing, testing, 1, 2. Okay. I was going to stream earlier, but it took me a while to reconfigure another computer. I had a computer failure basically, so I had to totally reset up all my streaming stuff on another one. And it had absolutely nothing set up for it, so it's quite a challenge to do it all from scratch. So we shall see how this goes. For some reason, on the PC I could get the mirroring really easily on the audio, so I could tell I could hear the music and my voice the entire time. And right now it's not mirroring, other than listening to a quick test stream video from YouTube. So I'm flying blind right now as far as the audio goes. So you guys can let me know if there's a problem with the audio balance. It sounded like okay when I started, but I'm not going to be monitoring it during the stream. I can't hear it. So that is now just need a couple seconds here to get a few things posted so people know the stream is on. I don't know why I picked that title. That's really silly. It auto chose a title for me, which is weird. Oh well, what can I do? Good old OBS. Voice is low versus music. Okay. Well, I boosted the mic level the maximum it can go with the OBS interface. Only thing I can do is drop the music a bit. Let me know if that helped at all. Just dropped it a bit. Okay. Yeah, I'm not feeling that image at all that it posted. Let me do a quick poster frame. It looks better. That will be much better. Okay. Also, you should be able to see my cursor in the window. Let me know if you can see that, because it's good for pointing some stuff out if I need to. Just quickly fix this poster frame because that is a really bad one. Whoa, what's going on there? That's bizarre. A bunch of guys just popped out of the cluster there. Interesting. So this is, I'm just starting with this because it's the first thing I found on the, got some diatoms there. There's it's about 100 power, so they're going alongside this, it's not a Celatoria, but it's another, looks like another blue-green algae or something. There's that giant rod, the segmented rod there. You see it's greenish from chlorophyll. It's also got a cluster of diatoms and other such things at the base there. And I thought these guys there, first I thought they were moving a little faster, but they're not. They're all pretty much diatoms there, with some other greenish bits mixed in the center of the cluster. I haven't looked at those yet. But let me just get this finished here, and I will investigate closer. I also had trouble with OBS trying to get my generative music thing I was using on a web page. Some ambient stuff that's basically free to use, but OBS on the Mac, at least the version I'm using, didn't like simultaneous audio streams for some reason. So I had to basically download a giant file and just have it play in the background. So I don't have any control of the playback like I did on the previous streams, but it should be okay. I did a quick preview of the audio file and the music was decent, so it's much better than silence anyway. Where's that file here? Modified. It didn't save it to the desktop. There it is. I found it. Come on. Copy that. And let's go back to my post. There it is. That's going to look better than the dumb poster frame it chose. This, the only option I had here was to create the stream right out of OBS and not do a lot of settings, so I had to kind of deal with the hand I was dealt today. Alright, enough of that nonsense. Let's start investigating. So these are samples I took the other day from the zoo I used to work at Palo Alto. Some of the water samples I took there about a year ago, well, actually more than a year ago. Created one of the best slides I've ever made, and it was just totally by accident. Just there were so many species and so many great specimens on one single slide from the sample. It was really stunning. Okay, so let's zoom in a bit. We can go up to 400 power here. And see, there's the colonial algae up close. It's got some really, really tiny, hairy structures kind of around behind it there to the sides. Those are really, really small cyanobacteria threads. They don't even have any color at this level because they're so tiny, but they're all related to bacteria. Basically, they're all prokaryotes. If you guys don't know the difference, prokaryotes are bacteria, cyanobacteria. They basically have a cell membrane, a cell wall, like a plant does. They also have no nucleus, so the DNA is in this kind of ring structure in bacteria, especially called a plasmid. They don't have a nucleus, so everything's floating around the cytoplasm along with the organelles, and so because a lot of them are fairly primitive, and even as far as organelles, they kind of really don't have a whole lot compared to a eukaryotic cell, which, as the theory pretty much still stands, it used to be called the endosymbiont theory. I forgot the latest name for it, but it's about a mutual relationship between a prokaryote and a eukaryote, and the chloroplast, which is in some ways similar to a mitochondrion with a different function, but both involving energy production, were potentially captured organisms inside of another cell, and they just became a mutual relationship and evolved together to become a single organism. And that's why you have mitochondrial DNA separate from the nucleus of a eukaryotic cell. Eukaryotic means it has a nucleus. Karyon means the kernel in Latin. Kernel meaning like a structure like a nucleus. And Those are the kind of the main examples of that stuff and main differences, but I could do a whole other stream on that if you guys want to see that. Oh, the slide is already starting to dry up because I left it on here too long. There's the wave front coming in. Let me fix the light. Lots of diatoms here though, and some algae specimens here, little colonial spheres there, or spheroids. And see that diatom is swimming there. It's a very active diatom. Got a few different types here. You have the kind that look like these little tapered rods. You have a few that aren't tapered. You go toward the middle of the slide right now. There are a few that are kind of more rounded ends. I can't even talk today. Rounded ends. That's a different species of diatom. You can see the yellow-greenish structures inside that contain the photosynthetic material. You can also see the oil droplets, those little spheres inside there that they have for storage. They don't, as far as I know, they don't make contractile. They don't make food vacuoles like other like ciliates and flagellates do. So I can't remember if the oil droplet's an actual vacuole or if it's an actual oil droplet that's just kind of, because it's hydrophobic, it's bound to a sphere inside the cytoplasm. But anyway, so that's lots of diatoms. Look at that big cluster of diatoms there. It's crazy. And some other algae in there giving it the green color. And there's a really long diatom there off to the side, forming like a clock hand there. And you can see the ridges there, like teeth of a comb, sort of, called a raphe. There's a nice little diatom right there in the middle, kind of moving around. There might even be the really common one called navicula, but I'm not sure. There's some that look like that. But yeah, tons and tons of diatoms. Even a huge diatom, that big rod there. So very cool. Very, very cool. Yeah, I got this water from the same day I went to see the sloth. In fact, the sloth footage, if you saw it, turned out pretty terrible. It was really choppy. I hate Verizon as a carrier. Once I get my phone situation straightened out, I'm not going to use Verizon anymore. I'm going to go back to Sprint/T-Mobile, because that stream was choppy for no good reason. I'm in the middle of Palo Alto with no interference around me, and it was just choppy. So I need to go back and get some more sloth footage. I'm probably going to take two cameras, use the phone for a live stream and then use another portable thing to record proper video to upload later, just in case I need the better quality. But yeah, sloth was amazing. Alright, so lots of little diatoms moving around, little submarines essentially. Slowly moving around, bumping into things. Always fun to watch. Since I left the slide out for a while, this may not be as did this pause? There we go. There was a pause in the transmission of the image. So now we're getting this slide is kind of now forming pockets because I left it out too long. Here's another cluster of, oh, that's a nice one there. So again, a lot of diatoms, a lot of various other, there. This one is cool. See, this colonial algae here, actually, this is one of those ones that's kind of loosely connected at little junctions. You see how it kind of got bent by the water pressure. I wish I could have caught that, because you would have seen that kink form right there. See, they are connected, but the water made it form a weird bend instead of being a straight line. I'm gonna fix the lighting here. We're gonna just right. You can actually see the outline of the cell walls there. So you can see they are connected. It looks like they're just touching, but they're actually connected, and that's why they have that bend. Kind of like those fold-up wooden rulers they used to have in the old days. It would fold up back into itself, and you put it in your pocket. They have these little kind of loose connections here. That kind of like, I guess, kind of like a hinge. Yeah, this other side did the same thing. But look at that structure inside there. Nice big photosynthetic structures in there. And some other stuff in there too. It's like a little Brownian motion making all the little things bounce around in there. Little spheres. Bouncy, bouncy. I might do a quick digital zoom here instead of, because I don't want to go to the oil lens right now, so let me do a quick digital zoom to improve that a little bit. You can see the bouncy particles bouncing around. Let me know if you can see that. Hopefully you can. Nice little structures there. This particular algae I don't know that well, so I can't really comment on the specificity of these given structures. I mean, they look like little chloroplasts in there as the spheres, and then the other green matrix material. It's chlorophyll, but I'm not sure why it's looking like that. It's kind of like a giant mat of cytoplasm kind of bound together. This one's kind of scrunched up in there. But you get that big space between the ends. It's like this weird air gap when it's really just some fluid space between the ends of each of the green sections inside the algae. We have this little water pocket that formed from the slide dehydrating from the evaporation. Made this guy bend right here. It's kind of funny. Weird, I'm getting a delay. The image isn't updating real time. It's almost like the camera is getting there you go. Now it's catching up. Let me know if you see that delay. I definitely see it on my end, but it could just be something weird on my computer. Getting a slight delay. When I just move this like that, it's usually real time between the microscope and the screen, but I'm getting a few delays between the actual scrolling and the image on the computer. So that's a nice little find, but since the slide is dried up mostly, I'll go to another one. That was pretty good for sample one. Sample one, which is this little kind of waterfall feature, goes into a little pool. It's pretty boring, though, sample I got the first day, so maybe I got lucky with this one. Let's try one more from sample one. We've got 4 samples total. Number 4 is the killer one, so I definitely want to keep that for last. Let's see what we get in this one. Let's see what we get in this one. Okay. Let's do a little dark field for a bit. Fix that lighting. What do we got here? Oh, nice big piece of... I got a piece of this last time. Nice lattice work there from this plant specimen. It's basically an algae that forms this lattice work. It's really interesting. Nice color, nice structure. I'm going to zoom in a little bit and see some detail here. because it's very 3D and thick, you'll see a lot of stuff out of focus as I go through the layers. Let me fix that a little bit so there's some better color and contrast. It's really beautiful. You get some hexagons and some pentagons. It's like a buckyball, like the Buckminster fullerene, which is also the pattern on a soccer ball, but buckyball is actually a carbon molecule, like carbon-60, and it has this repeating pattern of hexagons and pentagons to form a lattice work on a sphere. Named after the architect Buckminster Fuller. Oh, there's a guy swimming through there. That was cool. And he's off screen. Where's he going? Focus on him a little bit. See him actually cool. He's going 3D all through the ring structures are in and out, around. That's cool. Looks like it might be Euplotes. I like the little Euplotes guys. Those are cool. Let me slide this over a tab. Hopefully it doesn't cut off on the display. Let me verify. Okay, I have a little more breathing room to move it over so I can have my real estate back here. Okay, that works pretty well, I think. Okay, now I can see the chat window better. It's getting cut off before. Yeah, these are nice structures. I'm not familiar with this. This is a freshwater plant, and I'm looking mostly lately at marine specimens. So I need to look this guy up. But yeah, look at this if I focus through the Z. Okay, now I'm way out of focus, but it's through there, through the stuff to the back end, and then back through again. There's quite a bit of depth there. And little fun things swimming around through it. There's an air bubble. Is that a branch or something touching it? Yeah, there's some little things swimming around down there too. Looks like a little diatom maybe. No, it's a flagellate. It's moving too much. So again, more guys swimming through here. That wiggly swimming motion, that's a flagellate because that makes the body kind of wiggle as they whip the flagellum back and forth. There's all kinds of stuff in here. Look at that. Maybe I'll go one more level in. Let's see. Let's see what I can see here. Now it's really close up. This objective is not my favorite. I replaced it because I damaged one of the objectives being careless. And this replacement's not a good fit because it has distorted. It's like the wrong numerical aperture, so I need to actually get the correct replacement. Yeah, I'm getting that glitch again with the visuals are behind the scope. Come on, catch up. There we go. So there's a couple things swimming around through here. Ooh, and is that a big old diatom there? Oh, several of them. Look at that. These rods right there. Big old diatom rods. In a cluster. And all kinds of things swimming around them. Vibrating as they wiggle around. To go. Yeah, it's not going to go dark. It might go oblique lighting a little bit, but that's still not so great. So let's. Uh-oh, I got that visual glitch again. Come on, man. Why is it doing that? This laptop's an older laptop. It's kind of underpowered. I think it's just not keeping up. watch the stream and see what happened, but get the general idea. Let's go back a level. You can see the diatoms. They're actually in the back, so I have to focus in the back. Wait, no. That's actually the foreground. Yeah. Some of the 3D stuff is tricky sometimes. It's pretty cool. Alright, so I looked at that guy enough. It's more of a slide to check out, so actually I'll leave it at that power. Just do a sweep. Let's see what else we got. More diatoms there. Diatoms. So, piece of colonial algae. There's a chip in the glass. Look at that. Wow. Big old chip in the glass. Oh no, that's a thread with some water trapped. It's an optical illusion. What else we got here? There's an interesting cluster of cells. A little piece of tissue there from something. Definitely a big piece of multicellular something. Alright, let's cruise the edge. Sometimes things hang out at the edge. That's a chip in the glass right there. That's wild. Very concave. Okay. Go down the other side. Nothing. Yeah, again, sample 1 is pretty boring. That last slide was actually pretty good, considering sample 1 is not the most exciting. It's a cluster of stuff. Those little hairy things are getting a little cyanobacteria threads. Nothing. More cell fragments from a multicellular organism. Hey, look at that. That's a nice. So there's a little guy crawling on it too. Okay, that bear is close up. Actually, might see some activity inside. Let's go close up for that one. Let's see what we can see. Yeah, there's a nice detail there. That's definitely 3D. You can see it's curving off out of focus. I'm focused on the center point. And you can see as I focus through, you get the edge view. So it's definitely round and tubular like a bean pod. And all kinds of little guys swimming around it. Look at that. Behind it, around it, on the edge of it. Let's go to the bottom here and see what there's. Some more of those threads. There's a nice diatom at the bottom with the oil droplets and the photosynthetic region there. Very nice. More of those guys kind of swimming through. Let me fix the light a little bit. It's a little dark. There's that guy swimming through there. Hey, Teeny. How's it going? This is back to my water samples from the zoo from the other day. This is only sample 1, slide number 2. So I'm about to go to another sample. But this one's pretty good right now. For a while it's not so great, but a lot of stuff going on here. Nice diatom at the bottom there. That those little Navicula style ones. And some little spherical algae. There's some looks like a flagellate on the surface of this guy. Yeah, he's on the surface. Very slowly swimming around. Probably cheat a little bit more. I'm not going to go to the oil lens today. Let's do one more digital zoom and see if that helps a little bit. There you go. Let's get that centered. You can see that guy kind of creeping around the surface of this structure. It's like a ghost. There's a whole bunch of little transparent guys on here, and the big green thing is so large and overpowering, you almost can't see them until you focus just right. But they're just kind of creeping around on the outside of it. Shape, but it's just overpowered by the large green object there. I have to kind of defocus it so you can see the overall shape. It's kind of a weird tapered cell. Yeah, I don't think I've seen this guy before. And the little guys are too hard to identify. Just bopping around. Interesting. Oh, you gotta go? Okay, see you, Teeny. Sorry, it's probably late for you. Or during some food time or whatever. Yeah, digital zoom is just kind of lucky here. It's just it's the software digital zoom. So on the Mac, I'm having to use a Mac on this day because the PC thing has some problems. And the AmScope software was quite good for lots of little settings and stuff. This one's almost as good or similar. It's called the Pay version is Toup View. This is Toup Lite, and it works on Mac and PC, and it has a lot of the same settings as AmScope's viewer does. But this one on the Mac says a little bit different. A little less control with right clicks and on-screen buttons and stuff like that, but I can get around it. That's fine. But so digital zoom is all done from the software. The camera's a is it a 15 megapixel camera? It's the same one made by these guys all get like generic sensors and just slap their name on a thing. This is the AmScope model, 15 megapixel camera, same as the one sold by Swift, basically. They're a couple $100, a few $100, maybe almost $300, which is a hell of a lot cheaper than the DSLR, which is what Micro Hunter uses, which is really nice. And I'm actually saving up to get a DSLR for our astronomy channel that I'll also use for this, for the higher quality images on this stream. But for now, this camera's done quite well. My earlier streams were using a really cheap Chinese camera from, I actually bought it from Alibaba because it was the cheapest one I could find that was decent quality, and that was only about 5 megapixel, but the quality was pretty good. It had no editing features whatsoever. You hook it to one of these programs and you couldn't use the sliders, it just wouldn't respond to the software. So whatever mode it came in is what you're stuck with. And that was kind of crappy. So the exposure you could barely control. It was all exposure control from the scope itself, not from the software. No digital zoom, none of that kind of stuff. So this was an upgrade. And before that, I actually was using my iPhone bolted to the eyepiece for a while with those little eyepiece adapter clamp thingies, which are actually pretty good quality. iPhone camera is very nice. But again, it's just kind of hokey with it bolted to one side of my binocular thing, so I could look through one eyepiece and have the camera on the other eyepiece. And it just, iPhone gets hot after a while. It's not really, you got to keep it plugged in to charge so it doesn't go to sleep or die on you, and it just wasn't convenient. And if I still wanted to receive calls or test my stream or stream a podcast for myself while I'm doing a stream or recording some videos, it was just not really convenient. But in a pinch, if that's all you got, hey, I used that method for quite a while. So don't knock the what they call digiscoping, where we basically put a camera on an eyepiece. I've done that with spotting scopes for bird watching and for other stuff, and you can turn anything into a lens basically. It's quite cool. Just the quality is mixed results. I really like this guy. It's kind of a funny shape. I have no idea what it is. It's moving like a large, clumsy flagellate, or it's fairly large. But I can't see any details on it. Oh, right, there's the flagellum. I see it moving a little bit. But it's really slow. It's just kind of sitting there, probing around, hanging out. I don't know if the flagellum shows up on camera. It shows up on the eyepiece, but the camera always misses some of the details, unfortunately. That's another reason why I want to get the DSLR, because as good as those can be, even they will miss some details. Nothing beats live viewing a microscope with your eyes, if you can. Alright, that's cool. So that's an interesting specimen there. I think I already tried to focus through it so you can see the whole thing there. It's quite thick. And I'm not seeing any real activity in there. No cytoplasmic streaming, no kind of things moving around, not even Brownian motion. So, but I found this little guy, so that was fun. Let's go back a level. Let's see what else we can find. Okay. More junk. Whoa. There's a massive junk right there, like a microscopic stack of hay. Starting to get crystals forming now. As the slide is, oh, there's one of those little clusters, those little matrices of algae filaments. Really interesting. More diatoms there. Okay, so slide one was fairly interesting, but not great. Okay, that's a wrap on that. Yeah, you don't have to go nuts with the cameras. I was like looking at all these articles and YouTube videos and comparisons and stuff. I go, oh man, I want to spend a lot of money on a camera when I was starting my channel, as I was just learning how to use a scope properly again after many years. And, wanting to get some decent stuff to have for later. I could shoot all this. That's why I went to the live streams initially, was I was spending a lot of time shooting footage as I was exploring and going back and making prepared videos with narration and music and stuff. It's like, oh, this is so much work, and it's kind of fun to engage people and get people looking at stuff like they're there and having questions and stuff, and it saves me having to do extra time making all these extra videos. So it's a win-win for everybody to do these live streams. I love doing them because I can go back through my catalog like I did for the hardware jams video for the micro footage, and repurpose my footage for other projects. So win-win. It works out very well. Now let's go to sample 2. See what we can see. Sample 2 is from a tank of African cichlids, these freshwater fishes that are very colorful, very territorial. They fight a lot. They kill each other sometimes, depending which species you put together. A lot of competition in the lakes, like Lake Malawi in Africa. It's like down in Tanzania. And they're beautiful fishes, but they're very popular in the fishing, the pet fish trade. So there might be a lot of microscopic fish feces in here and stuff, as well as all kinds of little bits of weird things. It's mostly plant material so far. There's various fibers of things. Nothing too remarkable. Oh, now the camera's paused again. Come on. Don't do that. Oh, you're back again. Hey, Teeny. So now I'm sample 2. Definitely check back later if you can't stay for sample 4. I'll get to that last. That will be the best one, like it was the other day. Sample 4 is amazing. Alright, why is my... There we go. Now I get in my camera to respond. I also hear my Mac fan going nuts. I think the computer's chugging right now. It's having a lot of issues multitasking on some stuff. It's a much older Mac. It's like an older Intel Mac. But hey, I got the right OBS software. I got the microscope software on here. I got YouTube working perfectly. So it's not so old it's not usable. It's just a little slow. It only took me about an hour and a half to get it all up to snuff from scratch. So okay, so boring. So yeah, sample 2 sucks. Okay, so much for that one. Let's go on to sample 3, which is decent, I think. Memory serves. Sample 2 is not good enough to get more than one slide, and I don't like it. Oops. Okay. Slides are looking pretty good today too. I had to wash these a couple hours ago, so they're pretty clean. Alright, sample 3, what we got? What have we got here? Of course I put the covers up on at a weird angle. That's silly. Alright, let's just browse through. More debris. I got some water on the top of the slide there. Oh, there's a little guy. A flagellate bouncing around. But moving too much and too small to really study right now. Let's look for the bigger stuff. More debris. Nice little... That's a nice little piece of a plant there. It's a lot of cells. Something different to look at. Do dark field on this guy. There we go. It's nice. Looks like a little microscopic knife. Let's do push it one more just to see what the hell is going on here. Actually, it looks like it's a piece of fish skin with the scales overlapping, but it's very, very small. Not sure what that is exactly. Thought it was plant stuff at first, but it does have, looks like overlapping little tiny scales on it. But fish scales are even usually quite a lot bigger than that. But yeah, no, look at that structure there. You can see the little semicircles like it's fish scales. That's kind of cool. It's not a plant for once, or a single-celled animal. Crazy. Let's go back out because that mode. Oh, I got a delay again on the image. Come on, catch up. Catch up. Oh, Teeny had to take off again. Yeah, I'm going for a while longer because I got a few more samples. But not super late because I do want to get some sleep for once. So yeah, it's kind of a nice sample there. I like that. Whatever the hell it is. What else we got here? Let's follow the border. There we go. Got some filaments again, as well as some little tiny flagellates, some green algae bits. I wonder what that orange thing is there. What the hell is that? Some kind of tissue of some kind. There's a long ass diatom there. That long. It's at an angle, so I have to focus to see the whole thing, but all this stuff clustered around the diatom. And a little flagellate going crazy. Swimming through the debris. Busy little character. Alright, what's over here? Some more little things. And there's this reddish orange thing here. I'm not sure what that is either. It's very solid, whatever it is. And some more green sphere clusters there. Not so great, but interesting somewhat. Back to dark field. You can watch a little flagellate go to town again. Man, he's all over that thing. Very fast. What else we got? More filaments. Debris. More junk. Junk. Yeah, so sample 3 was pretty lame. With some nice little air bubble water mix. Looks like space art. I always like those little features. make several cool slides out of sample 4. It's a very good guess. Sample 4 is in... So sample 3 was a pond where flamingos and some other birds hang out. It should have been much better than that. But what's really cool is that water is being filtered into a catch basin. Like a pool filter kind of thing you'd have at home. On the that side thing where you get the leaves and stuff suck into the thing and then it gets filtered out and you take the leaf basket out and throw it away. It's kind of similar to that. And all kinds of debris and some of the little bits of shrimp that the things eat and other things get kind of caught in there. And it's just a smorgasbord of all kinds of really cool stuff, which is why last year it made one of the best slides I've ever seen and made. Kind of sound like that Steven Seagal quote, if you guys have heard that. I think it was David Spade was hanging out with him on the set of something. I think it was SNL or something like that, and he was backstage writing a script for his upcoming movie. And Steven Seagal comes out and announces to the room, "I've just read the best script I've ever seen, and I just wrote it." He's such a blowhard piece of garbage. I think these microorganisms are way more interesting and way more intelligent in some ways. There's a nice little guy there. What is that? Looks like a rotifer. He's attached and pivoting. Must be a rotifer. Sure enough, it's a rotifer. So they have this little foot thing that they can attach to debris, or in this case, maybe part of the glass. Can't tell from this angle. But you look very closely and you can see it's got the one part that's bouncing around. The end of it is, it's got this fuzzy moving parts. And it's moving these little lobes with cilia on it. Creating suction. Look at that suction being it's pulling in all kinds of stuff. It helps when I give it a little more brightness to the dark field. And you can see little particles being sucked in by the vortices created by this guy. I might go back just a tad, take some of the digital zoom out. Some of the fine details get messed up. And if you watch, see that particles there going toward him? So he's making suction. Now he detaches and swims around. They're basically filter feeders. So they're multicellular, about 1,000 cells roughly, but quite often are about the same size as like a paramecium or one of the larger single-celled ciliates. They filter feed just like clams and mussels do, and lots of other animals filter feed. In this case, bacteria and super tiny protists. Because they're not big themselves, but they're good size, but they're not huge, which is why a single-celled thing can be next to them and still about the same size. Like even a stentor can be bigger than a rotifer, potentially, and they're a giant single-celled tubular thing, like a trumpet. It took off. Where the hell did that guy go? Oh, there's something flying by. Alright, I'll find him later. Let's just slowly go through the clusters of goodies. See what we can see. But as you can see, clearly a lot more stuff on the slide already, and I just put it in. So diatom, we fixed it's blowing out like crazy there. Diatom in the middle is a rod, and all kinds of debris clustered around it. Same kind of stuff, filaments, little tiny flagellates. What we got here? More debris. cluster of air bubbles there. Making a weird shape. Alright, let's do sweep, sweep, sweep. There's that same kind of pinkish reddish color. That's a piece of the shrimp tissue that the flamingos eat in large quantities that gives them their pink color. Yeah, more tissue there. Something multicellular. Almost like an onion skin a little bit. Let's go back at a level because there are probably some things too easy to find on lower power in here. More light. Okay, what do we got here? More clusters of junk and debris. Clusters of junk there. Nothing standing out. Nope. Lots of colorful stuff, but nothing worth zooming in on yet. Okay, well, it's promising so far. It's just not seen anything that stands out as spectacular, but there's already a lot of stuff to look at on this compared to some of the other slides. Can't find that rotifer again. It went somewhere. Oh, what's going on here? Lots of little flagellates in this debris here. Something should go behind that thing. Oh, there we go. That's one of those nice, it likes to spin a lot. This one's funny. I haven't identified it yet, but it's, I see these quite a bit. In fact, I have a couple live streams where they stopped doing the contractile vacuole pumping for osmotic pressure regulation and basically popped after a certain period of time. But they are really fun. Pretty basic, like a cold capsule shape, fairly nondescript shape. But what's cool is when you zoom in on them and you get a nice view, especially with a dark field. There's so much stuff going on the cytoplasm. It's like a little snow globe. Kind of sparkly, kind of a lot of particles in there moving around. Really neat to look at them. Let's try to find it again here. Where'd you go? Where did you go? These little guys again doing their thing. I gotta find that big guy. Boy, he's really moving. He just took off. Could be anywhere. I have to go back to low power. There's this stuff they use in industrial stuff, food additives and stuff, called methylcellulose. Also used famously for the slime in the Alien movies, when the alien mouth opens up and the stuff drips out. Wonderful goopy slime stuff. There you go. Look at that beautiful little structure there. All kinds of stuff inside there, kind of shifting around. It's almost like a snow globe. They're so cool. I'm getting a better exposure there. You can see it. Let's do the full dark field with a better focus and you can see it, but now it's moving like crazy. It's spinning. There you go. So it's got a lot of colors from the stuff it ate. It's got some food vacuoles in there from various colorful things. How's that spinning motion? It's funny. But if I get the right exposure, they're just beautiful. Come on, buddy. Where'd you go? Let's go back a digital zoom a step or so. That'll help a little bit with the clarity, I think. Okay, where'd you go, buddy? Where'd you go? As soon as you get them doing that spinning motion, they'll kind of hang out for a while and exploit the fact that they're not going anywhere. I just have to do a quick sweep. Find it. Come on, man. Oh, there's that's another little squishy. This one's really cool. This might be a paranema. Very squishy little flagellate where the flagellum leads the way, kind of like a walking cane or like a blind person kind of probing the way as they walk. Alright, let's get that fixed. No, that's not working. Let's find it again. Where'd it go? It's right around here. There it is. Nope. It's debris. Let's go back lower. It is moving, so I gotta find it. Come on, man. Sometimes they just keep moving and they dart in another direction. You just lose them completely. Oh well. I was able to see it. I don't think I got caught on the camera very well, but I have footage of them from before. I can't find the big dude. The big dude just totally took off. And as you can see, the slide isn't that big. There's not a lot of places to hide. Whoa, now he's flying through there. Whoa, where are you going? In a hurry, going nowhere. It just kind of stops and goes back the other way. That's funny. Alright, come on. Big dude. Where's the big dude? I have to get low power again just to find that guy. He's a pain in the butt. He's a real pain in the butt. Sometimes he'll sit there and just spin for many, many, many minutes. You can just sit there and watch. There's a little piece of debris the shape of California. What do we got here? You might have gone off into the void around the slide. That happens sometimes. No, don't see it there. You really can't hide anywhere. It's pretty good size. I just have to scour the whole area. It also could be to hide behind some stuff. Sometimes they go tunneling for goodies. And they're like a cloaking device. There it is. Yeah, see, he's looking for goodies. I should have known. Should have known you'd be looking for goodies. Wow, so cool. And they spin a lot, so you can see the structure inside in 3D. nice. So another overall body shape is very plain looking. They've got a lot of stuff going on inside the cell. I will risk the digital zoom here, just to help out a little bit. There you go, lots of detail in there. Now if I get the lighting better, you can get that kind of crystalline look, and all the particles dance around inside like a snow globe. Just beautiful. It's too much zoom. Let's go back. So yeah, these are all the more beautiful ones. Not shape-wise or even the colors. The color in this case is coming from the food it ate. It's food vacuoles, but this just got a lot of... I wish I could just get one to stop for a few minutes. I could zoom in and just study the cytoplasm, but always on the move. Now it's hiding. This is how I missed it before, because it's in there hiding, looking for goodies. Totally miss it when it passed through. And this debris, look, it's just totally hiding in the bushes there. This is actually, this is only 100 power, but I got a slight digital zoom, so it's not a huge guy, but it's pretty big. It likes to squeeze in those little areas. They're just squishy enough to squeeze. And then he's gonna go around and see, spin again. There's a lot of this tumbling thing too. That's the thing that gives you the sparkly look. It's constantly spinning on its longitudinal axis. There's some of the sparkles. Beautiful. And they'll do that thing where they kind of squish around a corner and conform to the shape of the stuff. Yeah, this is good. It likes hanging out here a bunch, so I can just kind of chill here. This is the fun part of the stream, where I can just sit here and observe and not have to play the video game with controlling 4 knobs at once. Yeah, it's taking a break, looks like. Or did it take off and I didn't look? Oh, there it comes. It comes back. Just stay where it likes to be, and you'll find it every time. Let's do a little bit of... get some oblique lighting here, make them a little more 3D. That would be kind of fun. There's a little more 3D looking. Just have to fix the exposure a little bit. I'm just going to hang out here. You'll be back. It's really nice when you find a place they like to visit. Almost like a duck blind for hunters, just hanging out, waiting for them to come back. did you like that? Yeah, that was just screwing around. I know Kent usually goes on a roll playing all kinds of cool stuff, but it looked like he was just kind of tired and just relaxing. So hey, I said, hey, you want to hear some weird crap with some stuff I haven't played with for 6 months to a year? I had it all sitting here, so I thought I'll just set it up and have some fun. I do like the row at it. It is kind of like a poor man's Lyra 8 with only 4 parts, but I have a Lyra 8. I don't use it very much. I need to get back into that too. But the microcosm is just, it's literally cheating. It's so good. But there are tons of nuances in there. You can sit and fiddle with that for a while, as you saw I was doing, kind of trying to learn it some more and finding some cool tricks in there. And so it's really cool. You just feed either some nice stuff, it sounds nicer, or you can feed some really crappy stuff, and it still sounds great. Because it's sweetening. It's got, it's reverbs and delays and granular and glitch effects, smearing, filters, LFOs. Got different time controls on there too for things. It's got these repeating functions and ratchets, and yeah, it's a trip. I mean, it's a fairly expensive module, sound FX module, but it's really worth it. I think it was like $550 or something like that. I can't remember, and I had to wait for it to show up again. As you saw on the stream, it's hand numbered on the back, so they make them very small batches. It's actually got like an actual Sharpie serial number written on the back of it. So they don't screw around. They make good stuff. They make it to order, kind of small batches for stores and stuff. They don't have a lot of stock, so they do run out all the time. That's why I think like 4 times I tried to buy it, and it was like, oh, I'm ready. It's not there. I'll come back. And then I'm like, okay, I have to wait till I can afford it again as I'm buying other junk. Sure enough, I finally got it, and then I used it like 3 times and put it away. And then doing that hardware jam last week in San Francisco, I go, I need some effects. I'm tired of using some of these rack effects. They're nice, but the modular, rack effects are cool. But I said, oh, I got the microcosm. I should use that. And man, it was fun. It really helped the jam out a lot. It sounded great. So. It's pretty well built too, so secondhand should be fine. I mean, it's a nice stomp box, and the knobs are pretty sturdy. But again, you never know if somebody mistreated it, or if it maybe has. It's just a nuanced box. It'd be hard to tell if something was wrong with it. It's not so obvious like a typical FX pedal. So that's the one thing I'd be worried about buying a used one, because it would take me a while to even know something was wrong with it. Some of these sounds are really weird and crazy, and so that alone might make you want to buy a new one instead of a used one. I guess that guy's not coming back. Found some other little bushes and forest to play in. Hang out this one for a few minutes and see if anything happens here. But yeah, it's a killer thing, and I like the I have Source Ventress, which is a nice dual pedal. You basically have either 2 reverbs, or reverb and delay, or some weird granular stuff, or some other kinds of stuff, and either 1 at a time or 2 at a time, and it's a stereo pedal, and it's really, really cool. Also, the audio thingies from France, they make a thing called Dr. A, which has modular CV control as well as typical desktop features, and just a really cool set of reverb algorithms, like a black hole Strymon supermassive kind of thing, or weird ones, and glitchy noisy delays if you want, or beautiful smooth delays. It's a really nice little box. I've used it on some older YouTube videos from last year or year before, I think. And even the little Korg Mini KP is really fun. It's not the quality of all that other stuff, but it goes in your pocket, runs on batteries, and super handy. And has a record feature too, so you can, while you're using the effects and something going through it, you can record it. So it's a whole closed system if you want to do like a jam to it and record it. It's pretty cool. Alright, there's nothing here. so much more goodies I'm missing in this sample, so I'm going to make at least 2 more slides of sample 4. Sample 4 is very cool. Let's see what we can do. How's the music coming through on the back end? Ooh, nice. One of these guys again. Had these the other day. Oh man, there's all kinds of stuff on here. Look at this long. I've seen one of these before too. This is a really weird long ciliate, and I don't remember what it is. But they have this. They're very long, and they're. Damn, there's one of these too. Nice. Fat rotifer. See what I mean? This is the best goddamn sample around. I don't even know where to look, but I'm gonna get this guy because I haven't seen him for a while. These are really cool. Long ciliate with a clear kind of back side, which is kind of diagnostic, I think. And it's very flat. See how when it swims, it kind of swishes around. And you can see it rolling. It's got a flatness to it, but it is up and down a lot. I have to focus constantly. So it's a very worm-like ciliate. It's super cool. It's also got the sparklies going on. If you see it tumbling, it looks like a snow globe with all the stuff moving around inside. And the cilia dancing on the edge. Super cool. But this is one of those slides where where do I go next? There's so much to look at. I'm frustrated. Nice. But yeah, look how flat it is, and when it rotates, it kind of twists. It's not rigid, so it actually rotates as a solid rod. It kind of rotates and twists, and it's very much kind of a flat ciliate. Really cool. A lot of activity there. PAJM turns are kind of boxy. They just kind of move like a solid unit and flex just a little bit. So these guys are really floppy. Come on, man. A little more light on this guy, I think. I should stop moving so I could do more controls. There we go. There's a little oblique lighting, so it's got some 3D function to it. Very cool. And now it's got to focus. This guy's making me earn my keep here. This is a lot of work to keep in focus and to follow him. But it's worth it because it is a cool specimen. Damn, it moves a lot up and down, back and forth. Very sparkly. All that cilia dancing around on the right kind of light. Just has that snow globe look along with the some of the cytoplasm stuff moving around. It looks very sparkly. And this one rotates a lot. Man, it's really spinning on its axis. So active. It's also doing that arc behavior. Some of these things like to do these little arcs, going around in a circle. Other feature feature I want to do when I get the better DSLR camera is use a high quality slow-mo to shoot some of these guys, so I could really slow these motions down, study them, get some really good stills. Because these are moving so fast, the stills would be pretty crap on a typical camera. So cool, look at this guy. Now let's quickly go to kind of a normal light field, just for color comparison. I want to see the natural color. Nice bright field image there. So yeah, it's got a bit of a slight tint to it. It's not completely transparent and plain looking. It has a little bit of a tint, almost like an orangey reddish color to it, or pink maybe. So moving so fast I can't even tell. The back end is like a propeller. The way it's kind of bent, it uses it to twist like a little micro propeller. It's so cool. Very clever movement. It's got cilia for back and forth, and then it's got the twisted tapered tail end to also help it rotate nicely. Seems like it's also cilia doing that too, but it's a really interesting effect. I don't see ciliates doing this behavior. Damn, that's cool. Whoa, look at that cluster of diatoms there. That's weird. Oh, what do we got here? There's a rotifer again. And, oh, one of these guys is stuck here. Or is that the same one? That's stuck on some debris. Okay, he's cool, but he's really hard to follow, so got some good footage, I think. More debris. And rotifer's on the move. Go! There's a little tail there with a little foot. The foot has a kind of adhesive on there. It can glue to stuff, and as an anchor, so when it's doing the suction, it doesn't get pulled along with it. Because I've seen them use the suction to help them swim too. Whoa, what's that? That's debris. So that's kind of how it's swimming right now. It's using that suction to swim forward, but then when it has the foot planted, then it has leverage, and it can turn that suction on full blast and start pulling little critters toward it so it can filter feed. They're pretty funny. But you can see now, this is smaller in length at least than that ciliate I was just following. And this is multicellular. This is about 1,000 cells roughly. That other thing was one giant cell. So this micro world is a real crazy topsy-turvy area. Okay, that's some good rotifer footage. Let's see what else we got. This slide is just bustling. No amoeba yet though, and no gastrotrick, which I often find in this sample from that water. I still had, actually, oh, there we go. There's some of these guys again. These are really interesting. There's a lot of structure in these guys. These long trailing cilia behind them, kind of almost like fringe. They have this huge kind of oral groove thing lined with cilia. It looks like kind of like a comb going down the center of the body there. really weird shape. It's very asymmetrical. And they're super active as well. They're very hard to capture. A good piece of footage from. This one's not too bad. This one I had got squashed the other day when the slide was drying up. It was one of the ones that got squashed as the little water bubble was compressed down into a little bit as the slide dehydrated. These guys are really elaborate looking. I need to figure out what they are. I forgot. I think I looked them up once, but I can't remember. Don't see them very often except for this damn sample. I got it the other day and today. So, and you can see it has a nice concavity to it. It's kind of like a curved back and a concave surface when it rotates. Damn, this guy's fast too. Okay, I got a good footage of him. What else we got? Pretty good so far. Is that oh, Teeny's back. How's it going? So is this music mic combination working well? Again, I can't on this stream. I can't hear it because after I tested the initial YouTube video as it streamed, I did a quick adjustment. But the music is actually a long music file I downloaded that's looping generative music, ambient music, and I heard a couple clips of it. So most of it was pretty decent. But I can't tell what's doing now because again it's not feeding back into my headphones, so I have no idea what it sounds like. And if my voice is low, I can talk louder. I just didn't want to yell into the microphone. Had some good stuff so far. Let's see what else we can get. So far this, nice rotifer again. And another character behind that debris. It's like a little flagellate of some kind. Go rotifer. Let's try a little adjustment here. A little dark fields with some little oblique action. Make it pop. Very nice. Turn it too low. Okay, I'll turn it back up again. Again, I'm flying blind. I can't hear it in this new configuration, so I will turn the music up a bit. Hopefully that's not too loud. Nice little specimen. Look at that. All that crazy stuff going on inside his body. Very nice. I'm just gonna chill out here for a bit and see what this guy does. Get myself a break. Chasing those things down is really Kind of maddening. Oh, damn, it's already, God, it's almost 3 a.m. already. Whew, I've been going for probably 3 hours almost. I'm gonna give it, play with this slide a little bit more, do one more slide, and just for hell of it. This was supposed to be only about an hour tonight, but I was having so much fun, I kept going. So I'll try to do another short one tomorrow. I want to make the use of these samples because it's not easy to go there and get these zoo samples all the time because I had to have somebody help me get them. So yeah, I'm going to exploit them as much as I can. Get these samples to give me some nice results. So definitely do another short live stream tomorrow, possibly 4th of July since it's a holiday. Yeah, so tomorrow or 4th. Or the wait, today's the, yeah, tomorrow or the 4th. Yeah, today's the, oh, no, today is tomorrow. It's already 3 a.m. So tomorrow or slash today later or the 3rd, I'll do another stream. Getting kind of punchy here and getting tired, but I am having fun. Some of these rotifers have a really kind of a spiky tail that goes to this kind of a sharp spike, and some of them have like a long tapered body that ends in a kind of a bigger foot. So many different types of these critters. Lots of fun. Need a little more something here to give it a little more detail. I might even be able to cheat a tiny bit on this app. I forgot if it has a sharpen feature or not. That might help a little bit. Just for kicks. Yeah, there's not really a sharpen feature. Don't think. Just white balance, color adjustment. It's fine. Don't really need that anyway, but it would be a nice little bonus. I think the other one I was using on the PC had a sharpen feature if I really needed it, but it's okay. You can always take clips of this later and post-process it with Adobe Premiere and give it a nice unsharp mask to bring out some extra details. But not too much. I notice a lot of people when they sharpen stuff, the amateur video people, they really need to take it easy because you can just see over sharpened stuff really big time on YouTube and Facebook and all over the place. Even science stuff that's supposed to be, you're sharpening for a reason, not to just like make it look all crazy. But people just don't know what they're doing. So if I adjust this, you can see all those little particles in the vicinity doing a dance when it's pumping that little crown with the cilia. So all kinds of little tiny particles in that area. But it's not quite showing up on camera. Now the camera froze again. Come on, camera, keep up. There's the Mac. Yeah, I love this stuff, so this is not a chore for me. It is a chore to do those mad chases, the fast ciliates and stuff, the flagellants. It's kind of annoying, but I'm really excited about the end result, getting some good footage, especially of a rare one I don't see very much. So that's always a nice reward, although it is tougher when you're tired, and sometimes the sample is just, there's too much stuff, they're going up and down, so you have X, Y, and Z to worry about, plus the constant focus of the Z, because sometimes they go up and down in Z a little bit. Sometimes it's too much water depth, and it's really hard to do all that. Plus focusing and adjusting the, mostly doing all that chasing, plus also trying to adjust the contrast and the exposure is really a pain in the ass. It's essentially 5 controls, and you're pretty much only using 3 fingers on one hand and 3 fingers on the other hand, and you can't really, they're not all lumped together, so you can't really do 5 things at once, so it's kind of nutty. And also making up for lost time. I mean, I always loved microscopes as a kid, but my parents had money. They just didn't buy me stuff. I was never spoiled. I had to buy my own telescope when I was in high school, or just out of high school. I had to buy, my own microscope eventually. I have 2 microscopes now. I had a hand-me-down one I got from a lady who used to run the storage space place where I stored a lot of my books and other things, and she always was an interesting person to talk to, and she told me about some of the tenants and weird stuff they left behind. And one time they left behind a whole bunch of biological equipment, including some microscopes. So I ended up getting the exact model microscope we were using at one of my community college courses in biology. The exact old gray, 30-year-old microscope, but it was good quality. It just needed a little bit of work, so I took it to a professional to clean it, lubricate it, and make the action a little bit better, because the XY stage was a little bit slipping. He never quite got it fixed. It was pretty abused, but I was able to get some good stuff with that early footage that I haven't posted on here, with my iPhone kind of bolted to the eyepiece, like I said. But yeah, it's a lot of fun. So I've been catching up ever since then, which is, I don't know, probably off and on for 10 years. But I had a huge gap between all the stuff I studied in college and then going to professional work in IT and not having a whole lot of time for this stuff. And then working at a zoo part-time for many years, I got action. Got to able to volunteer some of my hours. I got paid, but I mean, I got to say, hey, can I move some of my hours to the Baylands when they need the help? And I can staff the building basically as a naturalist, doing public programs with microscopes and helping people with bird identifications and plant identifications and answering general questions. So that's when I really ramped up the microscope stuff. And eventually last year decided to buy all this equipment, because I was using a bunch of pretty good equipment at that facility, but I stopped working there back in, was it 2012, 2013, something like that. And then kind of stewed about it for years, looking at some of my old footage and looking at enjoying people online doing footage. I gotta get back into that stuff. I should just make a channel and do this stuff and just have some fun and share it with people and get people excited about it, because it is cool. And it was shocking to me, the people I worked with at that facility, as well as the city in general, that knew very little about this stuff, even though that's kind of their job. And here it wasn't even really my job. I'm just part-time having fun, and I made microscope ID charts for the local critters. I did some taxonomy stuff for them so they could explain how they're related, and they were like, oh, that's really cool, but I just didn't use it. I did a training class for some of the teachers, and these are ones who actually had more directed. I mean, I had a bachelor's degree in biology that I was going to go on to teach and do research in botany and zoology and stuff, but life gets in the way. You got to pay the bills, so I got my IT job. It was easy, and been doing it ever since. But these people who that's their career to do this stuff didn't really know what the hell, and weren't really that motivated to look, even though online has a million resources. So I was providing a service by making stuff that I was having fun with, and also educational tools they could use to pass on to stuff to kids. Because they look at something and they go, "Oh, that's a paramecium." It's like, no, that's not a paramecium. It's not even close. But they just gravitate to the common names that they know, common critter names that are kind of in pop culture. Even when sometimes there's no paramecium in sight anywhere, they just, oh, that's a paramecium. That's an amoeba. Amoeba is pretty easy because it's very specific looking, gelatinous, very much like our white blood cells. But they would say all these other things. I'm like, yes, that's a nematode, but that is not a nematode. That's a polychaete worm, and blah blah blah. So it's just kind of fun to provide some stuff they could educate the kids better with it. Because as you guys know, when you educate somebody poorly the first time, they'll never, most likely in their lives, correct it. Either they'll just keep remembering the bad information, or they will just or just not go any further with it and just assume what they learned was correct. Now you can really see it. I hope can you see the particles in the black field there? It's really filtering like crazy with this little vortex. It's moving so much I can't really zoom in too much, but it's in the eyepieces, obviously it's more, your eyes are more light sensitive. It's filtering little guys like crazy, but it's really hard to pick up on the camera. Now I got a camera stuck again. Come on, camera. Let's go. Now it looks like it left. Oh no, now it's back. He's back. But yeah, it's filter feeding like crazy on those little particles. So yeah, it's one of the big motivations for. Oh wait, stuff is crushing in. Is the water crashing in? Are we getting a slide drying moment? Usually the debris starts moving in like that. That's what's happening. No, not yet. That's weird. I wonder why it did that. And the camera's stuck again. I think the computer's having a hard time. I probably should have rebooted before I started the stream. That probably would have helped a lot because I hear the fan going crazy and the camera is getting stuck. So maybe I will make one more quick slide from the sample and shut this damn computer off so I can get a break. Because it's tired and I'm tired. Here comes magic number 4. Let's go. Fell. Come on. Here. What do we got? Okay, there's the edge of the cover slip. Let's see what we can see. Let's fix that exposure a little bit. Okay. So far, just some tiny stuff. Ooh, that's interesting. Nice collection of tissue and little guys cruising around inside. Let's get a close look at that. That's neat. Oh yeah, Vorticella. Beautiful. I had them on the last year when I got a water sample from the same thing. So they have these little filaments that they rest on. You can really see them clearly here with the as they retract to kind of protect themselves as they feed or as they feel kind of irritation. They also filter feed like the rotifers do. They have a little kind of a crown of cilia pumping away, and you can see stuff being drawn into their little cavity there. And the filaments they're on coil up like a little helix. So it's a very simple motion. It goes from a long thread to a quick coil. It's a super jerky motion. Almost looks like you're losing frames. It's so fast. And if you look carefully at the thread, it's a perfect little coiled, like a spring, as it retracts, and then it relaxes, and that guy's going way out there. And occasionally they'll detach, either because they had to or because they want to, swim around as a free-floating ciliate and reattach somewhere and start feeding again. These guys got really long filaments, though. They're way sticking out compared to what I'm used to. But you can see that stuff go flying by the head part there, as it's feeding. You see it sucking in, and if it's too big, it kind of bounces off and flies away. It's a rejected particle. So he's creating a nice little vortex there. If I can fix this exposure, you can really see it. See the stuff swirling around? That's great. I love this stuff. And sometimes a thing like that will cycle around over and over and over again because it's not bounced off hard enough, but it's constantly being rejected. And that crown of cilia is just a blur. It's going so fast, it looks like a little fuzzy line around the top of the cilia. But that's how fast it's going. It's even faster than the rotifer, it seems. This becomes this fuzzy line. Oh, great, now the thing is stuck. Okay, there we go. But you get these really cool, like, cycling vortices. This stuff is swirling, swirling, swirling. I've even seen, I have some footage from a stream from months ago where a bigger one of these guys is creating a nice vortex, and some ciliates are coming by and flagellates are getting sucked into the stream and bouncing off the body over and over and over again. It's like they're trapped. They can't get out of the wash like a jet engine. And there's some over here too. Look at this. Vorticella over here. Vorticella over here. They're all over the place on a nice long thread, little filament. I love the little dance. The vortices dance. Sample 4 comes through again. So cool. Can't wait to show it to my former coworker from the zoo. He's gonna trip when he sees this. And unfortunately, you can't see it in this magnification. But in the eyepiece, I can clearly see it because it's just clearer than the camera is. And there's so much activity inside the Vorticella's body, the cell. It's just all kinds of material cycling in there. It's, again, very sparkly looking, super cool. So if you can ever get a look at one of these under a scope, definitely try to, because there's so much detail I can't show you on this camera. I'm going to adjust the exposure a little bit, but it doesn't really help that much. Probably see a little more detail now. It's not overexposed. But they got dueling vortices. That one's got some going, the other one's got some going. And then they retract. Wow, they really retract. It's got some long ass filaments too. There's some of the longest filaments I've seen on a Vorticella. Usually they're like just a couple body lengths, but this one's like, looks like 6 or 7 body lengths. So I love these guys, the little wine glasses, I call them. Oh, camera's stuck again. Yeah, Matt, this is that same killer sample. Sample 4 is the magic one. Every time I go to that place and get stuff out of that trap, there's so much stuff, you know, because it's not circulating so much and getting washed away and stuff. It's just kind of this little tiny, tiny ecosystem inside this leaf trap. And it stays wet all the time, and there's plenty of stuff to attach to, and so it just creates this wonderful little environment, kind of a microclimate, microenvironment for these things to have the perfect conditions to just hang out there. They don't leave, and you know, they're safe from too much sunlight. There's constant stuff floating in, so they're getting new nutrients, fresh water, more oxygen. But there's plenty of reasons to hang out, and so I keep, every time I get some good stuff from that. It's really cool. I get a lot of stuff from the Baylands. That's great too, but it's totally hit or miss because you never know what you're going to get in a large, natural, open environment. But this is a tiny, confined kind of leaf trap thing. So I find some good. If the swimming pool at home in your homes didn't have so much chlorine in it to keep it nice to look at and nice to swim in, not get all green and nasty, that leaf trap would be amazing too. But there's so much chlorine in the pool, it's not very conducive to lots of life being there. Although sometimes you find salamanders and stuff in there because they're cool, they don't care. Yeah, these guys are so much fun. Now I can shift this because they moved a little bit, and they have dueling two sides of Vorticella. There you go. It's funny because there's two on each side, it looks like, so it's in stereo, double session quad. Although the other side's not as active, I'm not seeing the particle vortex as much. This bottom one they're just going crazy. I'm gonna have to probably zoom out then. I think my digital zoom is messing this up. Can't see both sides. Let's fix that. So now you can see it over there, but it's still not wide field enough to do it. It's wide field on the eyepiece, but the camera field of view is different than the eyepiece. But there's some good stuff over here. Look at this. They've got the double streams going now. I have to adjust the exposure a little bit so you can see it better. See the particles dancing around. Go, yeah, very quick. It's just so quick. It's just, even using naked eye with the, well, not totally naked eye, I'm using a microscope, but the eye without a computer or a camera involved, and it's so fast your eye can't get all the frames. So it's very jerky looking. It's nice showing the. Let's do a close up now. They've got some good, decent long shots. Let's do a nice close up on the heads here. So they've got some nice details. See the stuff moving inside the cell. There's all kinds of stuff streaming around, even a contractile vacuole going in and out a little bit. And if I focus through it, you can see that kind of ring of cilia at the top, like this little crown. And there's the thread right there. It's already kind of a twisted thread. You see, it's kind of tapered and twisted. It's flat a little bit. See the cilia doing that kind of wave as they're cycling around. Let's go down and look at this guy. There's the more standard like side view with the little crown above it. And kind of wine glass shape below it. If I get this just right, you might see the particles bouncing in and out. They're not freaking out as much as the other ones where they're retracting a lot. Let's go look at the ones down here and see what they're doing. There's one inside here. Filtering inside the piece of tissue there. And there's the guy in his little stalk. You can see the particle. It's got a funny angle there. It's kind of bent. Shading of it so it's not so washed out. Yeah, it was like these guys. We found these guys in biology lab way back in college. It's like, whoa, what the hell is this thing? I've never seen this before. So I was always on the lookout for it ever since. Just one of my favorites. And you always get a little cluster of them. They're rarely by themselves. You get like 3 or 4 on a little piece of debris or a stem of some kind or whatever, just hanging out, a little colony. So once you find one, you'll find a whole bunch kind of in the area usually. But yeah, there's all kinds of stuff going on in the cell. The cell is very busy with all this activity and all this feeding it's constantly doing. There's like the cell is just working overtime. It's just all kinds of packaging stuff going on with the little vesicles going from here to there. It's really cool. I just wish the camera would pick it up. The eye really picks up some great details. Contrasty there for some details, and I can really see the particle dancing around there. Cilia spinning around. Here, let's go back to slightly lower power. Let's get a nice beauty shot here. Particles dancing and all. I've never seen one's like kind of hiding inside a structure and one is outside doing it. It's just kind of, usually they're right about the same level. It's very funny. Cool. more sweep, because this was a very good slide so far. Guess that was it. Still pretty good though. The sample's nice too, because it settles after a while, and then it kind of gently tumble it so the stuff mixes nicely. And I noticed the water has a nice color to it, so there's tons of like little particles of good stuff that these guys would like to hang out in. So I know I'm getting some samples before I even take the pipette and get a little thing out of it. One more sample, then it's time to get some rest. Poor computer too. My computer's going crazy with the fan. One last one. That's how crazy this one gets. Diatoms, little flagellates, tons of little flagellates. Again. Hey, there's something on here at least. It's not a great slide, but you know, can't go wrong with the rotifer. It's an interesting structure there. That looks like it might be an antennae from one of the shrimp that those guys are eating. That's funny. Alright, there he is. Yay! He's also a fast one. And he's tumbling in and out of focus constantly. Come on, man, stop somewhere. This is the most active rotifer I've seen in a long time. Come on, dude. Oh, wait a minute. This isn't the rotifer. That's why. This is a wacky flagellate going nuts. Nice, but very hard to follow. Okay, where's Mr. Rotifer? Oh, there's one of those little Paranema things again, I think. Is it? No. Oh. Whoa. Oh, lots of little flagellates on this one. That one's nice, you can really see the flagellum stick out. Let me zoom in here. Some unknown flagellate, but it's cool with the... I can get the flagella image there. Nice. It's like vibrating, so crazy. Oh, looks like he's got flagella on both sides. That's why he's vibrating so much. He's just sort of wobbling, moving forward. He's just kind of vibrating a bunch from all that. focused just right, you can see it. Oh yeah, it's got dual flagellum, that's what it is. This might be... Oh, it's not Chlamydomonas, but there's several kind of single cell plants that have dual flagella out of one side. One is Chlamydomonas, we studied those in lab. This one doesn't quite have that look though. It does have the dual flagella. It vibrates like crazy. It'll crack up. Alright, let's see what else we can find. I had that rotifer before, but I don't know where it went. Can't find them. It's okay, I had rotifers earlier, so. Alright, well, last slide wasn't such a great closer, but I'm tired. Don't feel like making another slide. It's been a lot of fun. Thanks, you guys, for hanging out. As always, it's a blast. Got some cool stuff tonight for sure. The Vorticella was a star. And a couple of those other guys are pretty cool too, so definitely appreciate guys hanging out and commenting, asking questions. That's always fun to be dynamic. Hey, Kent. I didn't see you there. Were you there the whole time? I was just getting ready to shut down. I'll do another slide if you want to see it, Kent. I don't want you to miss anything. There's some great stuff earlier if you do the replay. Some good, really good specimens from the zoo. That's the water samples I got from the day I went to see the sloth. And this is, I did a stream 2 days ago with it, and then this is some follow-up because there's so much good stuff in the vials I didn't want to waste it. But, oh, you're not going to stay? Okay, no problem. Thanks for showing up, though. Definitely go back and check. There's some really cool stuff with Vorticella, these little wine glass ciliates that are on stalks. Filter feeding, and the particles are just swirling like crazy, and because they're rejecting the stuff that we can actually see, it's bouncing out, but they're actually eating the very small, like bacteria and stuff that are very hard to see. But there's all this cool vortices created by their motion, and it's just a trip to watch. It's pretty special, actually. So, and yeah, so I had some good stuff here. I'll probably do another one sometime tomorrow night. Possibly Monday, just to get the most use out of these samples, especially sample 4 from the filter box from the flamingos. That's the best, some of the best samples ever. It's really fun. So anyway, I'm sleepy. Take it easy, all. Thanks for coming by and hanging out. And my Mac has been giving us trouble since its fan's going crazy and the camera keeps stopping. But hopefully I'll reboot it tomorrow, make sure it's running very little material so it won't kind of have these glitches, and it should be a little bit smoother. So thanks again, all, and have a good morning, evening, or afternoon, wherever you may be. And I appreciate it. Good fun as always. And I'm going to end the stream. See you guys.